Well, thank you for the introduction. And hello, everyone. My name is Yuning Chen, and I'm currently a R&D manager at Sinobiological. And I'm very happy to be here. And I would like to take this opportunity to share it with you some of our work involving recombinant expression and application of influenza proteins for the purpose of universal vaccine development. First, I would like to give a quick preview of our company. Biological was founded in 2007. Our headquarters is in Beijing, and it is where I'm located. We have a few subsidiaries in China, as well as in Japan, and we also have offices in Frankfurt, Germany, as well as Pennsylvania, USA. So we're basically a little bit of everywhere. We are an expanding company, have over 500 employees right now, and our facilities are certified by international or domestic organizations such as ISO 9001 and CNAS. And our core business involves reagent development and sales, as well as customer services, which focus on recombinant antibody and protein expression, as well as antibody development. So that was a quick detour, and now I would like to focus our attention onto the main topic of the day, which was influenza and vaccines against this virus, or these viruses for that matter. So influenza is actually a old foe of the humankind. I think throughout the history, it has different strains of influenza has caused several outbreaks or pandemics worldwide. And even as for today, the disease burden of influenza remains. I think this is the data derived from a CDC survey, which indicates the number of cases as well as hospitalizations and fatalities caused by influenza. So even though it's an old disease, but it's still a heavy burden to the general health. And what's more, influenza is a zoonotic disease, which means it can spill over from animal to humans as well as the other way around, from human to animals. So a disease that maybe once a human problem, and because of the spillover event, it could spill over into the animal realm and cause other losses in terms of both economy and especially for the field of agriculture. So I think it is very necessary in this for us to be able to develop sustainable cures as well as vaccinations against such diseases to promote human health as well as essentially animal welfare as well. The influenza virus is a RNA virus, and it's comprised of its core material, which is the single-stranded RNA, and it encodes several proteins. Here is a graph shows the life cycle of the influenza virus. And because of the antigenic shift, the NA and HA proteins on this influenza virus can mix and match to create a variety of different strains. And this is one of the main reasons why influenza is such a potent enemy, for lack of a better word. So to take a closer look at the proteome of the influenza virus, we can see that nowadays the HA and NA are the main targets because of their abundance as well as their nature in antigen determination. And seasonal influenza vaccines has been developed and deployed on a yearly basis, and it can be comprised of different valencies, and they capture the essence essentially of the popular circulating strains from year to year. So now I would like to zoom in a little bit onto these recombinant influenza proteins and what can we use and how they can be used to facilitate vaccine development and also to to develop essentially seasonal vaccines, as well as a more quote unquote attractive idea, a universal vaccine against influenza. So in order to make these proteins, we use a traditional technology, which is recombinant protein expression. This technology has been around for, I think, over 4 or 5 decades at this point, and there are several well-established expression hosts that we can select from. For instance, we have a mammalian system expression system, such as the HEC293 cell lines and CHO cell lines. These cell lines are very efficient in producing the HA or NA of influenza viruses. While there's also an alternative so as to express these proteins in insect cells. The insect cells can also can produce a large quantity of both secreted and intracellular proteins. So, besides HA and NA, they are very good, useful tools also to produce nucleocapsid protein of the influenza viruses as well. And also, I would like also to mention there are E. coli. Of course, it's considered as the main workhorse of the biotechnology, and it's very helpful to produce nucleocapsid proteins of influenza virus and some other viruses. And also yeast is also another system that is available to produce virus proteins when necessary. Since the HA is the major target for influenza vaccine development, so I would like to take some time to talk about HA itself and also the vaccines that is based on these recombinant HA proteins. So the of influenza virus is a trimeric protein. It contains it's a single pass transmembrane domain, and it contains a large ectodomain and a small transmembrane domain and an intracellular tail. This HA is comprised of two major parts: the head region, as shown here in this angle, and the stalk region, as shown, where, as you can see, where the trimerization takes place. And from a phylogenetic point of view, the HA of influenza can be generally categorized into two major groups, group 1 and 2. Highlighted the subtypes that induced either pandemic or endemic in the past, as well as I highlighted the subtypes that are normally used for seasonal flu vaccine development. So this is how the HA looks like, and this is how it evolves to create different subtypes. However, within the same HA subtype, for instance, here the subtype of H1, throughout time, the figure may not be very clear when you see on this screen, but the study compiled several sequences from the H1N1 virus, the HA of H1N1 from different time points in history, and they found that the HA protein actually showed very well sequenced homogeneities. So, which means even though some of antigenic drift does occur, but since it's a very important part of the HA virus, of the influenza virus, it chooses to try to conserve most of its useful part as much as possible. And another study also pointed out that so we can take advantage of this conservancy in order to develop those quote-unquote universal flu vaccine. I think that this study was published last year and was elegantly done, where the stalk region of the H1 HA was taken and formulated into these hybrid HA molecules, and the people were immunized with these HA chimerics essentially, and they can develop antibodies against a different strain of influenza viruses. So this gives us hope that we might be one day able to develop a quote unquote universal vaccine to help deal with the current circulating variants as well as emergent variants. So, in order to facilitate such work, this is what we're doing at Xenobiological. So the one thing that we did is we have established a very extensive recombinant HA protein bank. So we have deposited recombinant proteins from previous pandemic and endemic strains, as well as we have a collection of vaccine HA strains for vaccine development. And also, we have established a very robust strategy for expression and analysis of these recombinant HA proteins. So basically, what we did do was we usually take the HA sequences and put them into a HEC293 cell system for initial screen. Some of the proteins are expressed smoothly and they can be purified and assayed for activity. But for other proteins, we might encounter some difficulties in their expression. So if that happens, we can either switch it into a different expression host, or we can just focus on the HA1, the head region of the HA construct, and express these recombinant form of these proteins in different hosts. And we've also established a relatively robust and high throughput hemagglutinin agglutination assay, as well as binding assays to facilitate the effort of obtaining biologically relevant and active recombinant HA proteins. So because we use different host cells to express these proteins, we did some study to investigate either the impact of the host cell system and what kind of impact of their post-translational modifications will have on the recombinant HA proteins. So in one study, we found that this is a H5N, the HA from H5N1. We found that a HEC293 is more prone to cleave the HA protein comparing to its insect cell counterparts. And by the agglutination assay, we can see that the protein with efficient cleavage seems to be better in terms of enzymatic activity. And for that matter, we also studied the host cleavage to see if it is absolutely necessary for the HA to be active in terms of its agglutination. And basically, what we did in this study was we took a HA protein from I think another variant strain. I think a H7 strain, and we mutated its furin cleavage site and removed the to eliminate the cleavage essentially. But we also find that despite this effort, the mutant and wild type showed similar potency in terms of its enzymatic activity. So basically, I think we can conclude from here that the expression host does have some impact in terms of its post-translational modifications as well as the enzyme activity. And so it will be good to have both systems available so that we can acquire different formats of the HA protein. But last but not least, I would like also to give a shout out of our virus research toolkit, a ProVir collection. So we have extensive collections of either recombinant protein reagents as well as antibodies against over 330 strains of different viruses. So we sincerely hope that this reagent bank could be helpful for your research and to also help altogether to promote human health. So thank you for your time, and I'll be happy to take any questions.